Oh, awesome. Well, welcome everyone. We have an awesome lineup of speakers for you guys tonight, and I'm really looking forward to some of the talks going on. A big reminder is our website. There's jobs posted up there, and so make sure that you check out what's posted, and then if anybody on here is hiring, make sure you put your jobs up there because it's a great resource for the community. But let's get started. I don't want to take up anybody's fantastic time for this mixer. So I'm going to randomly pick here. And so we're going to start with Ritu, and that will let her discuss her research and introduce herself a little bit more to us. So
okay, can you see my screen?
Yep, looks great.
Okay. So hi everyone. So my name is Ritu Chaturvedi, and I'm a final year PhD student at IUPUI, Indiana University, Purdue University in Indianapolis. And these are the areas of research that I'm interested in and that I've worked in. I'll start with protein chemistry. So I worked for around 5 years in protein chemistry, and this is a quick overview of the project that I worked in. So was my work dealt with the reconstitution of mutated GFP molecules. So for those who are not aware what GFP is, so GFP stands for green fluorescent protein, and it has a beta barrel kind of structure. So this is the structure of GFP, and it contains a chromophore that is encapsulated inside this beta barrel. Uh, it the chromophore consists of 3 amino acids. So these are the 3 amino acids. So my work was to mutate these amino acids one at a time to come up with GFP variants which are non-fluorescent. And then there were synthetic chromophores that were to be synthesized. And then the third step was to encapsulate those synthetic variants inside this beta barrel in order to come up with different variants of GFP with different quantum efficiencies. So for the sake of this project, I learned how to chemically modify a plasmid and the basic techniques of molecular biology. I want to put it out there that I did not know any biology before I started doing this work. I was a chemistry and math major, so it was really challenging for me. And I learned how to do gene cloning, protein expression, purification, uh, chromatography, ion exchange chromatography, gel filtration chromatography for the purification of proteins, and like the basic techniques like electrophoresis, agarose and SGS page, etc. Uh, Before I go further, I want to go back a bit and put it out there that I'll be talking about mass spectrometry and ion mobility experiments, but all the work that I'm presenting over here, these were the applications of these techniques, not like I'm not presenting any work that has to anything to do with the analytical part. Uh, in terms of synthetic organic chemistry, I've got quite a bit of experience. I've been doing it since my master's degree back in India. But for the sake of this presentation, I'll be showing the molecules that I've synthesized during the last 5 years. That means the molecules that I have used for my PhD thesis. So the first one that I'll start with is a thymine-thymine dinucleotide. So it contains two thymine moieties that are joined by a phosphodiester bond. So why I needed this? I'm sorry. So because for my PhD thesis, I was uh, trying to study the chemical reactivity of these UV induced DNA lesions. So what happens is when UV radiation falls on our DNA, it leads to the dimerization of nucleobases in our DNA. So primarily, these are the four kinds of lesions that are being formed. Out of these are the major ones. So CPD accounts for 90% of the DNA lesions that are formed in the human skin, and it is the leading cause of skin cancer. Now, I wanted to investigate the chemical reactivity of CPD lesion for two reasons. First, because I wanted to come up with assays for the detection and quantification of these lesions in the skin. And number two, if possible, we wanted to come up with some sunscreens that can prevent the formation of these lesions and possibly revert the formation of these lesions after they are formed in the skin. So that's a very ambitious and challenging thing, but that's what we tried. And in order to achieve both of these objectives, we needed to understand the chemical reactivity of this CPD lesion. So in order to do that, I synthesized dinucleotide models of this CPD lesion. So again, I started with TPT and then subjected that to UVB radiation in the lab and came up with this lesion. This is a dinucleotide CPD lesion. I've also synthesized CPD lesion containing a selective N15 label on one side. Now, this is more difficult and challenging than having a label on either side, because for this one, I first synthesized N15 labeled thymidine and then worked my way up to there, and the label had to be on just one side of the molecule. It does not have to be a symmetrical molecule. Like that, this was specifically required for one of my projects. I also synthesize a lot of oligonucleotides. So this is one of the representative oligonucleotides, which is a hexanucleotide, AATTAA. Now, I want to put it out there that this was not synthesized using a DNA synthesizer. I synthesized it manually using controlled pore glass beads, using the same kind of chemistry that is applied in a DNA synthesizer, but I could not use DNA synthesizer uh, for the kind of chemistry that I wanted. So I synthesized several oligonucleotides manually of different lengths. And after they are being synthesized, again, they were subjected to UV radiation. And then using reverse phase chromatography, I purified my lesion of interest and characterized and quantified them using UV visible absorbance. This molecule that I'm showing here right now, this has been the most difficult synthesis that I have done in like 15 years of organic synthesis. Uh, for this is a tetramer containing a selective N15 label on one side of the molecule. Now, again, for this one, I first had to synthesize N15 label thymidine and then convert that to a phosphoramidite. Now, this phosphoramidite, this particular step is really, really challenging because the yield of this reaction is really low. And this is highly moisture sensitive. And for the coupling reaction that follows after this, that used water. So after the coupling reaction, so this has to be, this is needed in excess during the coupling reaction, and I could not recover any of that. And I needed this molecule for a C13 NMR labeling experiment. So I needed at least 10 milligrams of this one. So it was a nightmare. I kept doing the same synthesis over and over again for like 6 months. So I've already established why I was making these molecules, because of the two objectives. First, to develop an assay, and second, to possibly develop a sunscreen containing that reversal molecule. So in order to do that, we had to investigate the alkaline reactivity of these lesions. Now, out of 4 of these lesions, the chemical reactivity, the alkaline reactivity of 3 of them has been already studied. So if you look closely, all of these lesions have a saturated pyrimidine ring. So it was a general hypothesis that when a saturated pyrimidine ring in any of these 3 lesions is subjected to alkali hydrolysis, the nucleophile attacks the C4 carbonyl group, leads to the formation of a hemiaminal intermediate that breaks this C4N3 bond, which further leads to DNA strand break. So now this is a known fact that when UV radiation falls on our DNA, it leads to the formation of these dimers, which further leads to strand cleavage, which is the cause of mutation and cancer. Now, for this particular molecule, all the published literature said that it is completely inert. It does not react even with very high concentrations of bases or acids. So there's nothing known as at all. Uh, Now, this was really surprising to us because a CPD molecule contains 2 saturated pyrimidine rings. So technically, even this should form a hemiaminal intermediate. If it's not showing any products, that probably means that it is not, these hemiaminal intermediates are probably not yielding any hydrolysis products and are going back. but there's no way to show this because I have been doing this. I kept doing this reaction by reverse phase chromatography, but nothing showed up. So we designed an experiment based on O18 labeling and thought of monitoring these hemiaminal intermediates if they are formed using time resolved, like using LCMS over a period of time. So what was done is this molecule was suspended in O18 labeled sodium hydroxide. And after every 24 hours, equal aliquots were withdrawn out of the reaction mixture and immediately frozen. So at the end of several days, all those aliquots were analyzed by LCMS. So this is the chromatograph at t equals to 0 days. So this is the normal isotopic distribution for dinucleotide CPD. This is the 545 is the mass of dinucleotide CPD in negative ion mode. This is M plus 1 and M plus 2. At the end of one day, we saw that there's a sharp increase in M plus 2 peak. That probably stands for the first O18 exchange. At the end of 5 days, the M peak was completely gone. And M plus 2 peak kept growing, and there was an M plus 4 peak that started coming up. That means there was a second exchange. At the end of 18 days, the M plus 2 peak also started decreasing, and M plus 4 kept going up. So this is the kinetic curve for the entire reaction, showing the rapid decrease in M peak in the beginning and slower decrease towards the end, M plus 2 peak, and then M plus 4 peak. Uh, So, based on this experiment, we could state with confidence that in a CPD molecule, there are two sites of nucleophilic exchange, and one is more reactive than the other one. Now, there were several more experiments that needed to be done in order to find out what that site of exchange is and which one is more reactive than the other one. But at least LCMS, this experiment based on LCMS, told us that, like, helped us establish that CPD is not an inert molecule. So it it does react. Uh, this like switching gears, I'll this is one of the other projects that I worked on, and I use ion mobility in order to solve one of my major problems for this one. So as I was talking about the sunscreen containing something that can revert DNA damage, so we were very fortunate, and serendipitously we found a reaction that helps us to repair DNA damage, uh, and help us to restore healthy. So this is a CPD lesion. On with the help of a reagent AX, it helps us to repair that DNA damage and restore healthy dinucleotide. Now, this was we were really excited about this work, and I personally wanted to characterize the entire reaction pathway. And I did find out that there were two intermediates that were being formed in this reaction, intermediate A and B, but like even after 2.5 years, I could not find out what those intermediates were. The major problem was the fact that both of these intermediates are highly sensitive to both moisture and uh, air, oxygen. So I was trying to monitor the reaction by reverse phase chromatography and LCMS, and every time I just saw the starting material and the product. So because none of the intermediates survived in those conditions. So then there was a point when Ian Webb came to our campus, and I realized that an ion mobility spectrometer that works under perfectly inert conditions. So if I can inject my reaction mixture inside this, there won't be any oxygen, and the reaction time is so little. So like in with less than 1 minute, I can get a chromatogram for my reaction mixture. So what I did is I did the reaction as usual on a Schlenk line inside a fume hood, and at different intervals of time, I took some reaction mixture out in a series that was equilibrated with argon and rapidly injected into the ion mobility spectrometer. So this is the chromatogram after at t equals to 1 minute, like immediately mixing the reagent X with my CPD. And this is what I saw. So I saw that there was no CPD, like the starting material was completely gone, and there was no product either. And the intermediates that we observed were exactly the kind of intermediates that we were looking for. So we were not able to find any evidence, but ion mobility served as a final piece in our puzzle and helped us put this reaction cycle together. So, uh, in terms of transition, uh, I'm looking for any field. that might be related to these fields of interest or any other field also, because you can clearly see I have no problem stepping out of my comfort zone. Uh, I don't have a lot of location preferences. If there is any good opportunity, I'll be there. So thank you FEMS for giving me this opportunity, and thank you everyone for your time. I'll be happy to answer any questions that you have.
Oh, thanks, Ritu. That was a great talk. So if everybody give her a virtual round of applause. So are there any questions? So I'll get started. So I didn't quite understand why you wanted the N15 label on the one side of your molecule and not the other side.
Oh, uh, so that's a huge project. I could accommodate only this one in 15 minutes. So should I'll share again.
Oh no, don't worry. You can
Okay.
just speak it.
Okay. So what happened is, using ion mobility, using LCMS, we could figure out that the reaction, that there were two sites of nucleophilic attack, but then I had to do the same reaction using C13 NMR spectroscopy. And then I found out that the site of attack was actually C4. Then we did not know which C4. So I had to synthesize this molecule because N15 is NMR active. So N15 will couple with C13 and lead to a splitting of that C13 peak. And the other side, there's normal N14, so that will not couple. So out of the two C4 carbonyl peaks on a C13 NMR spectrum, one will become splitted and the other one will remain intact. So that will tell us which one is 3 prime, which one is 5 prime.
Okay, gotcha. Anybody have any other questions? Well, if nothing further, let's give Ritu another hand and we'll head off to Charles now.
So let me share my screen real quick. I don't see my talk. One moment.
Yeah, that's always the worst. I always have so many files open, I can never find the right file to share.
Yeah, I got it.
There you go. Looks good.
All right. So I'd like to thank the organizers for letting me speak this evening. My name is Charles Mobley, and I work at the University of Mississippi in Dr. Sharp's lab. I started working in science at the University of Arizona, or I should say the Arizona State University, where I did my undergraduate work looking at organometallic complexes such as this here. And then I moved over to working at Vanderbilt University, where I did my PhD work. And I did a lot of membrane protein expression and purification. And I was able to actually purify many different membrane proteins, including 3 different versions of PMP22, which is shown here. And I actually was able to make the wild type in these 2 different mutations, which are disease mutations, and study those. And I studied them with several different characterization techniques, from NMR to fluorescence, as well as many that are not listed here. After finishing my time at Vanderbilt University, I started my first postdoc looking at expanding my ability to do NMR, my understanding of NMR, by going to the University of the University of Georgia, where I did protein structure determination using NMR. There's an example of that right here. And then I also worked on trying to figure out the best way to carry out NMR experiments, including the placement of spin label tags on membrane proteins. So if we put a spin label tag on here or one over here, what is the minimum number that we can actually put on and get all the information we need to actually determine the orientation of the alpha helices going through that membrane? And that was another one of my projects I worked on. And then I did additional membrane protein expression purification with connexins for a PO1 grant that unfortunately we only scored at 12% when the pay line was 8, which really was annoying. We were so close and just couldn't get that funding. But I did also serve as the president of the Postdoc Association at UGA, where I was able to improve my public speaking skills, my management skills and leadership skills, as well as my multitasking skills. And at the end of my time at the University of Georgia is when I met Dr. Sharp, actually part of this project. And he asked me to come with him to the University of Mississippi and start his new lab there. And in doing so, I started learning about hydroxyl radical protein footprinting, which is a technique where if you take a well-folded protein and you expose it to hydroxyl radicals, it labels the solvent-exposed residues on the surface of the protein, kind of like this shown for phenylalanine right here. And so you usually end up with a plus 16 or a multiplet of plus 16s or plus 32, plus 48. And in order to do this kind of work, excuse me, you need to be able to do LC-MS and bottom-up proteomics, which is what I was learning in the lab. And after figuring out how to do that, we started looking at ways to reduce the error in these types of measurements. And so what we've got up here is an example of data. We are looking at the oxidation event per peptide with either 2 microliters injected into the mass spectrometer or 5 microliters. And you see that with 2 microliters, you have very large error bars compared to what you see with 5 microliters. And that makes sense. But you also see that the actual oxidation events per peptide amount doesn't change significantly. So basically, we're just reducing the error. Now, at the same time, we coined a couple of new terms. Oxidation event per peptide coefficient of variation, which is basically looking at the amount of error in your experiment, and the total or the sum of the peptide intensity. And when you plot these against one another, what you see is that very low sigma pi, you have very high variation, and it kind of settles down to about 0.15 when you go to high sigma pi, except in cases such as these. And when we looked at these, we figured out that we're actually misidentifying oxidation events. And when we fix those misidentifications, we actually see that the data fits much better now. So this tells us that we have a way of looking for improperly identified oxidation and preventing errors from being propagated throughout our data. Now, this is not the only project that I've been working on while in Dr. Sharp's lab. I've also been looking at method development for non-trypsin digestible samples. These are samples that have regions of that are very basic and have a lot of arginines and lysines, and therefore they get cut into very small pieces usually. And that makes it hard for us to study those regions. So I'm developing methods to use other proteases, and that should be done in the next 4 to 6 months. But one thing that is finished as of this week is looking at detergent interactions with hydroxyl radicals. And this was spawned by some work done by Lisa Jones, where she was looking at whole cell HRPF experiments. And we were wondering why she wasn't seeing a lot of hydroxyl radical scavenging by the membrane. And the initial experiments that were done showed that membranes by themselves don't scavenge hydroxyl radicals very well. And that was very, very puzzling to us. So after thinking about it for a little while, we thought that we might use detergents as a mimic for lipids, and that possibly the problem could be the critical micelle concentration. And so if you're unfamiliar with this, what it is that for a lipid or detergent type of solution, if you're below the CMC, you have free lipids or detergents. In this case, I'm showing detergent. And if you get to the CMC, you still have free detergent, but anything that you add in after that, any lipid or detergent that you add in starts forming larger structures, such as these micelle, detergent micelles. Now, the CMC changes depending on your environment. So we needed a reporter assay. And the molecule that we wanted to use is ANS, which is a fluorescent dye whose fluorescent wavelength changes depending on which condition it's in. So it will move from fluorescing at one wavelength here to another wavelength if it's found inside of the micelle. The other thing I need to introduce is how we look at the amount of hydroxyl radicals that are actually present in our solution. So without hydroxyl radicals, adenine actually absorbs fairly highly at 260 nanometers, but then decreases in the presence of hydroxyl radicals. And if we take both of these pieces of information together and we, oh, it's not working. Something is not working. My screen is not progressing.
You want to jump out and restart it really quick?
Yeah, probably. Okay.
If you exit presenter mode.
Did you click on those little arrows at the bottom or not?
It's, I guess I can try that. There we go. Okay.
All right, we need a share screen.
Oops. Okay.
Perfect. Looks good.
There we go. So when we look at those two things together, where we look at the fluorescence that's occurring here, which is fairly stable until we get to this region here, and then it increases, indicating that this is probably the region of the CMC. And then we look at the change in the absorbance of the adenine, which decreases until this region. Basically what it's telling us is that since it's constant afterwards, that absorption is due to the free detergent actually scavenging the hydroxyl radicals, because you don't see any change after that, because all of the additional detergent actually goes into these micelles. So that's what I've been doing with a lot of the HRPF work. But what are some of the other applications that I could bring to an employer? Well, there are several different ways that HRPF can actually be used. So there's traditionally protein ligand binding experiments, including epitope mapping. There's conformational changes. So you can look at changes in higher order structures, say going from a phosphate buffer to a citrate buffer, although you've got to be careful about that because citrate will scavenge hydroxyl radicals. Or you can do things such as what Lisa Jones has been developing recently, with doing work inside of live cells or multicellular organisms. And we've also been working in the SHARP lab to see if we can't determine a way to use HRPF to do structure determination of proteins. And that's something that I've also been working on, but don't have near enough time to go into right now. So with this in mind, and with my background in NMR spectroscopy, cell culture, LCMS, homology modeling, FPLC, HPLC, and UHPLC, what am I looking for as far as a job? Well, I'm looking for an industry position, preferably something in the senior scientist level. I would really like to be able to use my skills with HRPF or other structural biology skills. However, I have no problem doing protein expression or purification. And you might be asking, why does he want to switch from academia to industry? Excuse me. And the reason is because even though I really enjoy helping students in the lab figure out the experiments, and I really enjoy helping them progress in their careers, I want to be able to help society as a whole as well. And I've recently seen some experiments where they've used HRPF to actually make things like, or I should say not make, but test things like antibodies. They're being targeted to proteins such as this BAM-A and the membrane of E. coli. And those antibodies bind here according to the HRPF data, and they inactivate this protein and actually end up killing most of the E. coli cells. And to be able to do work like that, I think would be very, very, very fulfilling for me. And a little bit of a change, which allows me to get more experience. Now, the other question is, where do I want to actually do my work? And that's a fairly easy question for me. the San Francisco Bay Area because my wife is going to be starting her new position there in a few months. Now, with that, I would like to acknowledge all the people who have helped me with this work, especially Joshua Sharp, who's my current mentor and boss, all of the members of the Sharp Laboratory, including Anter, who is actually working for me in the cell culture lab, and my former boss, James Prescott of the University of Georgia, who taught me pretty much everything I know about NMR. And with that, I'll take any questions.
Oh, if everybody can give Charles a big round, that was great, Charles. So do we have any questions for Charles?
You can't let me get off easy.
No.
I would just say that was a really great, interview presentation. You ticked a lot of boxes there. Nice job.
All right, well, I guess I'll have to let the next presenter go ahead then.
Yeah, you got off easy. So
Sure.
we'll, so one more hand for Charles and we'll hand it off to Gayathree. And hopefully I didn't slaughter her name too much, but.
Gayathree?
Gayathree.
Yeah,
there,
it's
I
Gayathree.
screwed up the beginning. I knew I did.
That name struggle is so real.
All right, I think I'll go ahead and share my screen. Hi, everyone. I'm Gayathree. I'm a final year PhD student in the Center for Systems Biology and Molecular Medicine in Yenneper University, Mangalore. So to just talk about my academic journey, I completed my bachelor's and master's in engineering from Anna University and Sastra University. And during my master's, I qualified 2 national level examinations, one being the graduate aptitude test in engineering, and other one is the national eligibility test for the award of fellowship for pursuing PhD. So in that NET exam, I qualified with a national rank of 33, with which I activated my junior research fellowship in Vision Research Foundation. Over there, I was working in deciphering the role of early growth response 1, a master transcriptional regulator in the context of diabetic retinopathy, where we proved its role in human microvascular retinal endothelial cells, primary culture, and also in diabetic retina, we confirmed its role as pro-angiogenic. Then I transferred my fellowship from there and I moved into Yenneper University, where I was a senior research fellow. And my fellowship tenure got over with the national eligibility test. So I was awarded another fellowship from Department of Science and Technology, India, which I'm currently being funded by. So this is where I'll be discussing about my current work in Yenneper University. Over here, you can see that this is a university which is situated in Bangalore. And our lab, Center for Systems Biology and Molecular Medicine, we have an Orbitrap fusion tribrid mass spectrometer and a QTRAP 6500 mass spectrometer. And my work being, I have been handling the QTRAP 6500 mass spectrometer for over 2 years, where I was trained in metabolomics and multiple reaction monitoring, that is targeted analysis. And my focus was mostly onto targeted and untargeted metabolomics, where I was working on in-house and collaborative projects. So my role involved from sample prep to data acquisition and analysis. Since I'm in the final year right now, currently I'm involved in data analysis and helping my juniors in maintenance and handling of the mass spec. So today's presentation will be on my PhD research, wherein we plan to evaluate the neuroprotective functions of Yashti Madhu, that is a traditional preparation from the roots of Glycyrrhizae glabra. So one of the main themes of our laboratory is molecularizing traditional medicine. So on that basis, this was my PhD topic. So to see the neuroprotective effects of Yashti Madhu, we were looking into the, in a Parkinson disease model. So we used IMR32 cells, which are actually neuroblastoma, and we differentiated that into neuronal cells using retinoic acid. Upon differentiation for 9 days, we treated it with rotenol, which is a mitochondrial complex 1 inhibitor, which is a classical model of Parkinson disease. And we also treated it with the Glycyrrhizae glabra extract. So here I will be mentioning it as GG extract. I'll be using the scientific name rather than the traditional name so that it's easy for you all to understand. And After treatment, we went into multiple, integrated omics analysis. So we went for quantitative total proteomics, comparative untargeted metabolomics, targeted metabolomics, and quantitative phosphoproteomics. So before we went on to the cell culture experiments, we first wanted to validate the licorice root powder that we obtained from the Ayurvedic practitioners. So licorice is nothing but Glycyrrhiza glabra. When we looked into the untargeted metabolic profiling of this licorice root, we were able to find glycyrrhizic acid and glabridin, which is very specific to glabra. We also found other licorice metabolites such as licorice saponin G2 and licrutin apioside. So this we were able to identify them at fragment level, because one thing in metabolomics is that you need a very good database to which you can map all the MBIZ features onto their respective metabolites, because usually most of the database, they are restricted to the precursor identification, but we had to go ahead with the fragment level identification so that we have a confident catch on whatever metabolites we're saying. So we didn't stop with that. We also took all the metabolites that we identified in it. So we identified around 1,500 putative metabolites from licorice root, other than the other 4 that I've just shown. And we also predicted what are all the probable proteins that could interact with these metabolites. So on identifying these targets, we identified about 107 human proteins that can probably react with them, interact with these licorice metabolites. And then we did pathway analysis. You can see over here that we identified several signaling pathways such as receptor and non-receptor tyrosine kinases. Then we had MAPK signaling pathways, including ERK1 and 2 and P38, and also PI3-AKT signaling, and then cell cycle pathways. So taking this into consideration, so we had a layout in front of us once we did this licorice metabolites on what we had to expect in our cell line models as well. So first is the model validation, wherein we had to see if rotenone is working in our cell line model, and is the GG extract, is it conferring neuroprotection. So for the cell culture experiments, we took an aqueous extract of the root powder, and we optimized the concentrations of rotenone and the extract. So we were treating with 100 nanomolar rotenone and 200 microgram per ml of the extract for about 48 hours. It was a co-treatment experiment. And you can see that from the live dead cell staining assay, rotenone shows more number of dead cells, which is stained as red with propydium iodide, compared to the co-treated cells. So we also checked for the expression of this pro-apoptotic proteins, that is the initiator caspase, cleaved caspase 9, the effector caspase, cleaved caspase 3, and the downstream cleaved part. We can see that they are all increased with rotenone treatment, while it is decreased with the co-treatment with the extract. So we confirmed that the extract actually prevents rotenone-induced apoptosis in the cells. So once we confirmed that, we went for the quantitative proteomics approach. So over here, we took 2 independent biological replicates, and the data was acquired as technical triplicates. So this was a 4-plex TMT experiment. We identified around 144 proteins that were altered by rotenone, significantly based on the FDR cutoff, and we took a fold change cutoff of 0.8 to 1.25. And the treatment with the extract actually restored about 84 proteins, and additional regulation of 4 more proteins. So over here, oh God, yeah, this is the slide, I'm sorry for that. Yeah. So the cod plot over here shows what are all the proteins that are dysregulated by rotenone and what are all the functions it is involved in. So you can see that it is predominantly most of the proteins dysregulated by rotenone are involved in mitochondrial functions, including those that are involved in oxidative phosphorylation, that is the electron transport chain, and also the tricarboxylic acid cycle. And also in protein aggregate clearance and protein degradation, that is E3 ligases and autophagy pathway was also affected. And endoplasmic reticulum functions were affected, and proteins involved in cell cycle regulation and DNA repair and replication. So when we also did the pathway analysis of the proteins that are restored by this extract, which you can see on my right side, we can see that some of these proteins that are involved in these pathways were actually effectively restored. So we concluded that from our quantitative total proteomic analysis that rotenone predominantly affects the mitochondrial function, which was efficiently restored by the extract co-treatment. So keeping with these things in mind, we went with comparative untargeted metabolomics. So over here, we had the same 4 conditions over here, and we use the protein concentration as the sample volume normalization. We use the protein concentration, to take the equal amount of samples. And then for metabolite extraction, it was quenched in liquid nitrogen, and we extracted it using a triple solvent method, which has methanol, acetonitrile, and water. And then we also spiked in an external standard, which was epicatechin, because epicatechin is a plant metabolite, which we will not be expecting in our cell line. So we spiked in with that, which was used as an external standard for the normalization experiments. And we used MZ-MINE analysis for extracting the MBIZ features that has MS information and alignment across all the groups. So for the untargeted and targeted metabolomics, we took 3 independent biological replicates, and the data was acquired as technical replicates for them. Alignment of all these 36 files resulted in 1600 MBIZ features that was acquired in both positive and negative modes. And we also mapped it with the human specific database, which was generated from human-seq and HMDB. We mapped around 400 metabolites in positive mode and 200 metabolites in negative mode. So when we did the PLSDE analysis, you can see that rotenone shows a very unique clustering compared to the other 3 groups, showing that there is a differential metabolite landscape when we give it rotenone treatment. The differential metabolite analysis gave us around 400 MBIZ features that were differentially regulated across both the positive and negative modes, of which only 98 were actually mapped to any known metabolite from the database. So we took all those 98 metabolites and we did pathway enrichment analysis. Over here, we got very interesting leads that are inconsistent with the quantitative proteomics data. You can see that glycerol phosphate shuttle, which is involved in the generation of the NAD plus in the glycolysis step, which is transported into the mitochondria. And then we had citric acid cycle, which is taking place in the mitochondria, and cardiolipin biosynthesis, another lipid biosynthesis, which is important for the mitochondrial function. Warburg effect, which deviates the pyruvate from entering into the citric acid cycle. Instead, it converts the pyruvate to lactate. And we also got metabolites, that is the electron carriers in the mitochondrial electron transport chain and glycolysis. So when we cumulatively put everything, we know rotenone is a complex 1 inhibitor, and we're also getting proteins and metabolite pathways that are involved in mitochondrial function. So we were like, very well, why don't we just go ahead for MRM analysis of the citric acid cycle? So we did the targeted analysis of the metabolites in the citric acid cycle. Over here, these are just representative transitions that we took. So over here, this is the surrogate standard of epicatechin. And we can see that citrate and isocitrate, which are actually isomers, you can see that we were able to resolve them at different time points at 2.1 minutes and 1.6 minutes. If you look at the transitions also, the fragments were picked such that there are no overlap, so that we won't have any issues when we are identifying these fragments in our samples. So I've also added a representative transition for the succinate. So every metabolite that we monitored in the in the citric acid cycle had minimum 3 to 5 transitions, of which one we took for one of the fragments was taken for the quantitative analysis, and the rest were taken just as qualitative. So over here, you can see that rotenone is actually disrupting a lot of the metabolites involved, almost all of the metabolites involved in the citric acid cycle, while the treatment with the extract actually sufficiently restored alpha-ketoglutarate and succinate levels. And we have to see that these 2 levels are very important because, these 2 steps, they are involved in the generation of the electron carriers, that is the NAD plus and FAD plus, which in turn take part in the electron transport chain. And since these 2 metabolites, that is succinate and alpha-ketoglutarate, are already lower, their levels are low, meaning that the electron carriers will also be expected to be going down, their levels as well. And rotenone also inhibits complex 1. So there is overall deficiency in the presence of the electron carriers. And we expected that the efficiency of the oxidative phosphorylation in terms of generating the ATP was lesser when we treated with rotenone, which was effectively, or let's say partially restored by the extract treatment. So when there is deficiency in ATP, the thing that happens immediately in the cells is the stress sensors that come into play. That is the energy sensors. So one such energy sensor that comes into play under energetic stress is AMPK1. So when AMPK comes in, AMPK and mTOR, they're actually having opposing functions. When there is lower level of ATP, AMPK is active and mTOR complex 1 is inactive. So we looked at several proteins that are in the mTOR complexes and in AMPK, and also the upstream proteins, but I'm showing only representative a few of the Western blots here. You can see that mTOR is phosphorylated and AMPK is also phosphorylated. But one interesting thing that we noted was Raptor was also phosphorylated. Now Raptor is a direct substrate of AMPK1. And when Raptor is phosphorylated, it actually leaves the mTOR C1 complex. The role of Raptor in the mTOR-C1 is as a substrate recruiter. Only when Raptor is present, the substrate binding will take place with the mTOR-C1 complex, and mTOR can in turn phosphorylate its substrate. Since Raptor leaves the complex because it is phosphorylated by AMPK1, mTOR-C1 is inactive. So our consensus is because of the lower ATP levels, which is as a result of rotenone treatment, AMPK is active and mTOR-C1 is inactive. We need to confirm that as well. That's why we went into looking for ULK1. ULK1 is the initiator kinase in autophagy. So this site at serine 757 is the site for mTOR, which is downregulated, and the serine 555, which is the site of AMPK1, which is increased. So from this, we can conclude that yes, AMPK is active and mTOR-C1 is inactive. So there is activation of autophagy, but one could argue that autophagy is actually good in neurodegenerative diseases, because the thing that we see in neurodegenerative diseases is the aggregates of aggregates of proteins in the neurons. But the problem here is when autophagy is activated when there is already cellular stress, there is definitely mitochondrial and oxidative stress because of rotenone. And in such cases, when there is stress-induced autophagy, the process does not complete, thereby leading the cells to apoptosis. So we were able to see that the cell death is actually happening because of mitochondrial dysfunction, lesser energy levels, which leads to AMPK activation and mTOR-C1 inhibition, and further activating the autophagy, thereby which is not able to complete itself, and it is leading to the death of the neuronal cells. So then, keeping these things in mind, we wanted to see what are all the kinases that plays a major role in this neuroprotective functions of this glycyrrhiza glabra extract. So we did quantitative phosphoproteomics here. We did it in early time points such as 2, 5, 15, and 30 minutes. We wanted to capture the kinase that first comes into play as and when we treat the extract. And if you could recall from the slides, that is the initial slides wherein I showed that the metabolomics of the Licorice root, we had several pathways such as ERK pathway and P38 MAP kinase and AKT. We were able to identify these kinases which is projected in the human kinome map. So some of these pathways we have identified, MAPK pathways, and we had even cyclin-dependent kinases which regulate the cell cycle. So we were able to conclude that yes, the glycyrrhiza glabra extract is conferring neuroprotection and how it is doing. So from the integrated multiomics analysis using metabolomics, quantitative phosphoproteomics, and total proteomics, we were able to see that rotenone dysregulates the rotenone-induced dysregulation. It involves energy stress as a result of dysregulation in TCA metabolites, which leads to stress-induced autophagy. And there is also evident mitochondrial stress, which in turn leads to cellular apoptosis. But wherein, when you treat it with the glycyrrhiza glabra extract, it is able to restore the functions, thereby preventing the rotenone-induced apoptosis. So that is it. I would like to acknowledge Council of Scientific and Industrial Research India for the NET scholarship, and Department of Science and Technology for my scholarship that I'm currently availing, and Biotechnology Skill Enhancement Program, which offered fundings for our lab and infrastructure provided by our university. Also, I would like to thank Dr. Ravi Shankar Parvaji and Hari Krishna Panaji, who are the advisors in the traditional medicine upfront for us. And I would like to thank my mentor, Dr. Professor T.S. Keshav Prasad, and my thesis supervisor, Dr. Prashant Kumar Modi, and Mr. Altaf Najar, my colleague, who acquired my proteomics data, and Mr. Santosh Behra, who created the metabolomics database for the searches. So my future career plans. So I have experience in metabolomics and mass spec handling, and also data handling for proteomics and multi-PTM studies. I am also trained in handling integrated omics data and arriving at conclusions for them. And with the background of cell and molecular biology, I would like to explore my further postdoc in either of these technology. I would also love to do work on spatiotemporal analysis of protein dynamics. And my location preference, there is currently none. And this QR code, it takes you to my LinkedIn page if you're interested. So I'd be happy to hear from you. I'm looking for opportunities now for postdoc. Thank you.
Thanks, Gayathree. So any questions for Gayathree? Everybody, please give her a virtual hand.
There was I think there was a slide in the beginning where you were showing the untargeted metabolomics of the plant. and then you
Yes.
connected up those MS2 fragments to like potential human protein targets.
Yeah, actually, from whatever the metabolites that we identified, we took the SMILES ID of the metabolites, and we have this binding database. When you submit the SMILES ID, it gives you the probable protein interactors of those metabolites. So it is giving a scoring based on the metabolite structure. It could be the exact match, or a structurally similar metabolite could be interacting with that protein.
Okay, and is that database
Yeah.
predicted protein compound interactions, or is it proven ones?
No. Most of the interactions are proven, but when we get a similarity score, it is a predicted one. Only when there is an exact match, we can say it's proven. So we give a structural cutoff of 85%.
Got it. Thank you.
Yeah.
Any other questions?
No, but kudos to Gayathree for waking up at 4:30 in the morning to join us today.
Yeah,
I was going
she
to
needs
say
some
that.
strong
Can I see
tea.
your graph?
Sorry.
I like some of the graphs you had. I thought they were really neat.
Okay, thank you.
Pathways. They always confuse me, so I really like the schematics.
Okay.
Yeah, so let's give Gayathree one more hand, and we can do a quick breakout networking section after this. So great job. And I really want to thank all of our speakers tonight. I learned tons of stuff. So that was definitely a fun hour of science. Do you have a question, Amber? Or
Yeah, I was wondering if before we go into the breakout rooms, if we could do a quick poll, if anyone here is doing the mentoring pods, if you've had your meeting already, if anyone had any feedback in terms of, do we need to get any more information out there? Did everything go smoothly? Everyone's okay? Just nobody has any needs? Okay, awesome.
no thumbs down out there.
I know. Well, we don't even have a thumbs down reaction in those reactions. Maybe this is like the laughing one, but we can pretend it's crying. So you can do that one if you're having problems. Okay, go ahead, Erin. Thanks.
Yeah, definitely ping Amber on the private chat if you guys have things that you need for the mentoring pods. But I'm glad that ours was really fun. I thought Vinny's actually in mine, so that was really fun. But yeah, let's break out for 10 minutes of networking. And so I'll break you guys into breakout rooms of about 4. So your question today, since it's almost Halloween, is what was your favorite Halloween costume? And especially since that was brought up earlier in the discussion. Or if you haven't dressed up for Halloween, what's your favorite candy that you wished you could get for some Halloween this year? So let me break you guys all out. Let's see, we have 21 of us. We'll do 4 participants per room, but yeah, I'll see you guys back in 10 minutes. So off you go. Have fun. hey, Wei, how's it going? Hey, Wei, if you're talking, you're muted.
Hey.
Hey, how's it going?
Good. Very busy. I'm busy with preparing for a submission for work that I've been working for like 4 years, 4 to 5 years.
Oh,
so
wow.
details to finalize. So look at my eyes.
Well, you're lucky because you might have missed the time zone because you're an hour late. So they're in breakout rooms right now, but we're finishing in like 2 minutes.
Oh, okay. Great.
So then you'll have more time than you were even expecting.
Okay. Well, I just noticed I missed the last week's the talk from the editors like John and Joe and Vicky.
Yeah, that one actually turned out really well. We recorded that one though, so we'll get it online so people can listen to it. But that one was really fun.
So I guess I've missed a lot of fun.
Oh no, you're doing fine. It's a busy time. I'm gonna call these guys back.
I like your background. I just watched this movie with my son like last week.
Oh yeah, it's one of my favorite ones. I love it.
I like. Oh, I also like the one they go to university or.
Yeah, I know that one I think is hilarious. Yeah, it's like Monsters University or something.
Oh yeah, Monster University. Yeah.
Yeah, no, I loved all the Monsters Inc. and Monsters University. Those ones I think are hilarious. Especially Billy Crystal and I forget, I think it's John Goodwin and as Sully. They're hilarious.
Oh, yeah. I watched the Chinese version because my son he he wouldn't English, so.
Oh, that's awesome. Yeah, no, they're some of my favorites.
Are we still allowed to talk about the